Last speaker is Katie from Stanford. She's a rising fourth-year PhD student in chemical engineering with a focus on medical diagnostic devices. She's been involved in InterVarsity since her first year in grad school, and she's currently on the steering committee at Stanford. She says in her free time, when she has free time, she enjoys hiking, skiing, and crafts. So now she will be sharing with us about where she sees God in disease diagnostics. So take it away, Katie.
Thanks, Anna. Yeah, guys, so as Anna mentioned, I'm Katie, and I am a fourth-year grad student at Stanford. And today I'll be sharing with you my square-inch story, which I have titled DNA Wars, Attack of the Mutations. So when I say this title and when I talk about DNA and mutations, for some people, the first thing that might come to mind is some crazy sci-fi stuff like cloning. So for example, things like the clone army in the Star Wars movie Attack of the Clones. But I'm actually here today to talk to you about something quite different, and specifically lung cancer, and how we can find better ways to test it, and how my work in this field interacts with my faith. Just to give a brief outline of what I'll be talking about, I'm gonna go, pretty nerdy on you guys with all of my bio stuff, so just get ready. I'll give some basic background information on the central dogma of biology and mutations in DNA, and then talk a little bit about the specific gene that I'm looking at, epidermal growth factor receptor. And then I'll give some background on circulating tumor DNA, and then give a brief overview of the research that I'm doing, which is mutation detection with magnetic sensors. And after that, I will talk about how my work informs my faith by giving an analogy related to the central dogma, and then kind of talk about two implications of this analogy. And then, we'll have discussion questions. So I wanted to start off with a brief review of some important background concepts for the non-bio people in the room. And bio people, I'm sorry, I know you've probably seen this like a million times at this point. So the central dogma of biology summarizes the typical flow of information in living things, starting with DNA, which is the longer-term storage of genetic information. And then this can be replicated over and over. The DNA sequence then gets transcribed into mRNA, which is more of a temporary copy of that same genetic information. And then after that, the mRNA gets translated into a protein, which carries out a specific function in the cell, so like forming structures in the cell or catalyzing chemical reactions. Ideally, these sequences would be passed down through this chain with perfect accuracy, but in real life, obviously, that's often not the case. So when DNA bases get copied incorrectly during replication, we get mutations in the sequence. And these mutations propagate down to the resulting mRNA sequences. And then depending on what the exact mutation is, this could cause anything from no effect on the resulting protein to a drastic change in the protein's ability to function properly. One example of a protein that can get drastically affected by certain mutations is this one called the epidermal growth factor receptor, or because that's quite a mouthful, we call it EGFR. EGFR, which is shown in blue here, is this multi-part protein. It has one part which sits outside the cell, and this part picks up signaling molecules like this pink one here. These signaling molecules are floating around outside the cell, and this part of EGFR kind of picks that up. And then it causes this part of EGFR that sits inside the cell, as a result, to trigger this complex set of downstream signaling pathways, which make all sorts of things happen inside the cell, including proliferation and dividing into other cells. So normally this works, pretty smoothly and things go well inside your body, but unfortunately sometimes mutations can occur in this particular part of EGFR. And when these mutations occur, that disrupts EGFR's normal functioning and causes it to actually signal for too much cell proliferation. And when you get too much cell proliferation, that eventually leads to the development of cancers, such as non-small cell lung cancer. When a patient first presents with lung cancer, they often don't know initially what mutations they have that are causing the growth of that tumor. And eventually, if they do get tested to figure out what mutation they have, if they find out that they have one of these EGFR mutations, there are actually specific targeted drugs that they can take, which will actually be often more effective and have less serious side effects than the more generally used chemotherapy and radiation therapy. So ideally, a patient would want to know that information, but traditionally testing for these mutations in a lung tumor requires you to actually extract the tissue from the lung, which obviously is a very invasive process and is also very costly. But in more recent years, people have found an interesting thing about tumor cells, which is that they actually shed quite a bit of their DNA. So when the cells either break open and release the DNA, or when the cells just secrete the DNA out through the cell membrane. And when they shed this DNA, it gets released into the bloodstream and is known as circulating tumor DNA, or ctDNA. All cells in the body release DNA into the bloodstream to some extent, but tumor cells often release relatively more, and especially in the later stages of the cancer. Now, because this DNA gets shed into the blood, we can actually take patient blood samples. We can centrifuge them to separate them by density, so we can separate out different components like the blood plasma and the red blood cells. And when we do that, we can actually extract this mutated tumor DNA from the plasma, and that actually presents us the possibility of having a much easier, less invasive way to test cancer patients for relevant mutations. So that's where we get into my work. So detection of circulating tumor DNA in blood samples is what I focus on. There are a lot of different ways to do this, but my lab specializes in doing it with these magnetic sensor arrays on these little printed circuit boards, which are about like this big. They're pretty small. And we pattern a bunch of sensors onto these chips, and then I can go into a lot of detail about how these work and what technical challenges are involved, so feel free to ask me about it afterwards, but I'll just give the very basic overview here. If you remember from your biology classes, two DNA strands can bind together when their sequences match up. So like when an A matches up with a T and a C matches up with a G. We basically make use of this complementary binding to pick out DNA strands from the blood that have the specific mutations we're looking for. And then we use these tiny magnetic beads, which are called nanoparticles, to attach to our DNA of interest and give us a signal readout in our magnetic sensors. The final signal that we measure from the magnetic sensor then tells us whether the mutation we were looking for is present or not. So if the mutation is present, a lot of nanoparticles will get stuck on here, and we will be able to detect a signal from them, so as shown here. And the other thing that's interesting is that because we have an array of these sensors, as I mentioned earlier, we can actually test for a different mutation on each one, which gives us the ability to look for a lot of different mutations in a single blood sample. So essentially, with this kind of test, we can figure out what mutation a patient has to help them select the most effective treatment. And because it's a less invasive test, we can also actually continue testing them on a regular basis to monitor how they're doing over time, to see if the amount of ctDNA is decreasing, which might signal a decrease in tumor growth. We can also see if a new mutation popped up that makes them resistant to the current drug they're taking. As I mentioned earlier, a lot of different methods for this kind of testing exist, but our goal in working with these magnetic sensors is to make tests that are cheaper and more portable so that they can actually be used in more resource-limited settings. So how do the things that I just talked about and the things I work on inform my faith? There's probably a lot of different answers I can give to this question, but for the one that I'm going to focus on today, I want to go back to that central dogma that I talked about in the beginning, where the DNA gets copied and replicated and then transcribed into these more temporary mRNA copies and then translated into the proteins, the functional parts. Someone recently suggested to me a really interesting analogy to this that I thought would be cool to share, and maybe it's a bit cheesy, but just bear with me. So in this analogy, we can think of God as being like the original DNA, and then we humans as being the copies of that DNA, because we were made in his image, and we're obviously also a lot more flawed, a lot more mutated, if you will, versions of him. Our thoughts and our words are like mRNA. They're temporary copies of our current state of mind. And finally, our thoughts get translated into our actions, or our proteins, the actual physical manifestation of what's in our hearts and minds. At the risk of stretching this analogy too far, there are a few insights that it brought to my mind, and I'm going to be talking about those now. So the first insight is the importance of proteins. DNA is really cool, and I mean, that's what I spend all of my time working with. And I often forget that at the end of the day, proteins are the things that actually carry out most of the cell's physical functions. Apologies to the bio people in the room for the generalization. I know there's a lot of exceptions to that, but in general. In the example that I talked about earlier, the EGFR mutations in tumor DNA are very helpful as markers that we can detect, but ultimately the messed up EGFR protein itself is what actually drives the growth of the tumor in the patient's body. In the same way, I often need to be reminded that it's important to not just know in my head how God wants me to live my life, but to actually live that out and show it in my actions. As was said in Luke 6:43 to 45, for no good tree bears bad fruit, nor again does a bad tree bear good fruit. For each tree is known by its fruit. The good person out of the good treasure of his heart produces good, and the evil person out of his evil treasure produces evil, for out of the abundance of the heart his mouth speaks. And so the fruit that we bear, or the proteins that get translated from our thoughts, is what others will know us by. And not only do our actions reflect on our own minds and hearts, but they can also affect how others view the rest of the church as well. Knowing the impact our actions can have, it's important to be intentional about the way we live out our faith. Another implication of this is that it's just as important for DNA copies to exactly match the original DNA in order to ensure that the resulting proteins are made correctly. So it's important for us to conform ourselves to the image of Christ so that we're able to produce good with our actions. So given that, how can we practically go about conforming ourselves to the image of Christ? Well, in your cells, copies of DNA often get checked for mutations in order to get those errors fixed. As shown in the figure here, it takes a whole group of different proteins, which are called mismatch repair proteins, working together to accomplish this. As you've heard about EGFR mutated lung cancers, obviously this proofreading machinery doesn't catch every mutation, but it does catch a lot of them, and without it, our cells would be in much worse shape than they are now. In the same way, it's really important for us to have a community, a group of people around us who help keep us accountable by noticing these mutations when they pop up and helping us to fix them together. The verse that this brought to mind for me is Ecclesiastes 4, verses 9 through 10. Two are better than one because they have a good reward for their toil. For if they fall, one will lift up his fellow. But woe to him who is alone when he falls and has not another to lift him up. When we fall and we make mistakes, we want people around us who can pick us up. And just as importantly, we should also be there for others to help them when they're struggling. So with that, just to summarize, the main takeaways that I mentioned were, one, that the fruit we bear, our actions and how we live out our faith in our everyday lives is what others will know us by. So DNA is important, but the proteins are just as important. And second of all, that it's very important for us to surround ourselves with and maintain relationships with a community of people who have our backs and can pick us up when we make mistakes. So yeah, I think we'll distribute the discussion questions later once we go into breakout groups, but just as a heads up, the discussion questions from this are, what are the proteins that you, like the fruit that you bear in your everyday life, and how do these actions affect the people around us, and how do they reflect on our relationship with God? And then also, what are the communities that we surround ourselves with who are our mismatch repair proteins, and how do we maintain relationships with them?